Society and Indian Society of Chemists and Biologists. He is passionate about quizzing, reading, writing, and also he has done a critics in poetry both in Tamil and English. He has reviewed many books such as Selfish Gene, The Double Helix, Gun Germs and Steel, The Planet of Viruses, Freakonomics. And he has also created awareness among school and college students about dangers of substance abuse, rise of antibiotic resistant, and he has given career guidance to the students. Thank you very much for the opportunity given to me. Thank you all again. Over to the speaker.
Thank you, madam. That was a lovely introduction. I'm very humbled. Thanks a lot. Thank you for the head of the department and the principal and all the faculties of the Bharati Women's College for giving me this opportunity to give a talk on bioinformatics and its role in drug designing and drug development. Thank you once again for this proud privilege of being a speaker at this eminent event. So, can I start the session, madam?
Yes, sir.
So, is my presentation visible to you? Is the presentation visible to everybody?
Yes,
Yes,
visible,
sir.
sir. Yes, visible.
Okay, sure. So we'll start the talk on harnessing the power of bioinformatics in drug development. So as the name suggests, we are going to see how this field of bioinformatics is very useful in the field of drug development, especially in this lockdown time when the world is in hunt for suitable medication against novel coronavirus and also for a vaccine. So we will see how bioinformatics can be really useful in this field of computer-aided drug designing and how computers are going to help in, are actually helping in the drug development process by cutting down the cost and also cutting down the time. So before we go on to the talk, we must know what a drug is. So drug, broadly speaking, can be any substance that is absorbed into the body of a living organism and it alters the physiology of the organism. Physiology means normal body functioning. So in the field of pharmacology, drug is defined as any chemical substance used for the treatment, cure, prevention or diagnosis of a disease or used to otherwise enhance the physical or mental well-being of a person. So drug is very, very, very important. So back in the 1800s till the 1990, actually till the year 1988, the year I was eventually born, the field of drug development was always called as forward pharmacology, which means that drug development was done by serendipity. Serendipity means luck. So even when Alexander Fleming first discovered the penicillin during the World War II, it was because of luck that he was able to identify a fungal organism called penicillium, which was able to inhibit the bacterial colonies. So until 1988, all the drug development process was mostly from natural sources or in the later 1900s through synthetic process, but it was all trial and error. You try a drug, it will work out or it might not work out. If it works out, then you put it for clinical trials. If it doesn't work out, you throw it out. But only after 1988, we started having the concept of rational drug designing, more so because of the work of George F. Fletcher, one of the eminent biologists of the 19th century or rather the 20th century. Today, the drug development process is generally synthetic and there is unlimited possibilities, and it is a highly standardized and tested mechanism, and there is tight legislation and laws across the world which are regulating the introduction of a new drug into the market and also how drugs are given to patients, the dosage, etc. So first we need to understand a little bit of economics before we go to talk about bioinformatics. The pharma industry is highly capital intensive. So to give you an example, one particular drug called Viagra, which was introduced by Pfizer company in the 1990s, it earned more money than all the iPhone sales so far, which means that the money that can be made through the pharma trade is immense. But you must also remember, if the money is going to be made in a large amount, then the capital that needs to be put in must also be very high. Even a very large company like Pfizer, which has a net asset value of about $300 billion, not 1 billion, 2 billion, $300 billion. That company has only 11% of the market share. The entire pharma industry of the world is about $3 trillion worth. The Indian economy itself is only $5 trillion worth. So the entire pharma industry is about 3/5 the value of the entire nation of India. So big is the pharma industry across the world. And if I ask you to guess how long it will take for a drug to come to the market, the actual answer is it may take about 10 to 20 years for a drug to be developed and brought to the market. A lot of students are here participating in this lecture by me, and most of you would have been born somewhere around 2000. You are all millennial children mostly, or you might have been born in 98, 99. So many of the drug development process which has started in the year that you were born are yet to make it to the market. So long it takes. And for a drug to come to the market, it typically costs around $300 to $500 million. And this money is really big. And investors always expect high return on investment. Even for a movie like 2.0 acted by Rajinikanth, if the budget is 400 crores, then the producer will eventually expect at least 600 crores of distributor share so that the producer goes home happily. Similarly, in any business, here in this case, the pharma industry business, if somebody is going to invest $500 million and going to spend 20 years on a drug development, they would definitely want to make money in billions. So the drug development process in olden days was a very long time process. There was more failure and the investment was more. Today it is a more efficient process and less investment is made because of bioinformatics. So every year in the United States of America, $324.6 billion money is spent by the taxpayers on buying drugs. So much money is involved in the pharma industry. That's why I'm emphasizing it again and again. But only 22 drugs were approved in the year 2016. If you compare it to 1961, more than 3,400 drugs were approved that year. The reason is that the pharma industry has become more regulated recently and more drugs are being attritioned. Attrition means dropped out of the pipeline. So if you take the drug development process, there are 5 stages: target identification, target validation, lead identification, lead optimization, and clinical phases 1, 2, 3, 4, and then you go for the market launch. The target identification and target validation process are carried out by the biologists like you and me. And the lead identification, lead optimization work are done by medicinal chemistry and biochemistry people, whereas the clinical phases involve clinical trials to healthy volunteers and then to patients who are suffering from the disease, and then it will go for a market launch. So the first 2 steps, target identification. Target identification means this drug, when you consume it, which protein or carbohydrate or lipid in the body is this drug going to bind and cause a physiological impact. That is what is called as target identification. I have to find out where this drug is going to bind and what this drug is going to do to the body and what side effect I might get. All that needs to be understood by target identification. So there are multiple techniques today available, like genomics, like next generation sequencing techniques, or microarray, or yeast-2 hybrid technology, or other RNA technologies like serial hybridization of RNA. These technologies are employed in target identification process, where the protein of interest or the glycoprotein where the drug is going to bind to is identified. The second process is called target validation. Target validation means you are validating whether this target is good or not, whether your drug will properly bind to this target or you are having a false positive result. Identifying that is called target validation. And then you come to the lead identification process, where the medicinal chemistry people will find out which particular chemical entity, it might be from natural source, from plant source. You're all botanists here, so you must be interested in this field of science called pharmacognosy, which means trying to identify natural compounds which are going to have pharmacological properties. So it might be from natural sources, or it can be biosimilar molecules. Biosimilar molecules means synthetic compounds that have a structure like the natural compound, but maybe with better efficacy. And then they do a lead optimization. Lead optimization means they are trying to modify the structure of this naturally occurring compound or the synthesized compound in order to suit the structure of the protein that the drug is going to bind to. So always remember, there are two entities in the drug process, very important entities. Two very important entities. I'll tell you what it is. One entity is what we call as ligand, Ligand, ligand. The other one is called as, one second. Ligand is the drug molecule that we are going to use to target the protein. The protein target is called the target. So two words you must remember. One is called ligand. Second terminology is called protein target or target. Mostly the target would be a protein. Sometimes it might be a carbohydrate, or sometimes it might be a lipid, or sometimes it might be a glycoprotein. Glycoprotein means mixture of carbohydrate with the protein. So you must remember that very importantly, these four stages are where bioinformatics are used. But in the clinical trial phase, where the drug is given to healthy volunteers to test the adverse effects, and then given to diseased people to test the efficacy of the drug, nothing can be done. The time frame or the cost cannot be reduced at all, because this is highly regulated by laws. So you might have seen in many movies that drugs are fraudulently tested in poor people, helpless people. You might have seen it in multiple movies, but that is all bogus stories. Even in India, the legislation, the laws are so strong that no innocent person can be subjected to a drug trial without his consent and without his understanding. The laws projected by the Indian Council for Medical Research are so strong. The bioethical laws are so strong in our country that no poor person or no underprivileged person can be exploited for a drug testing. So I just wanted to share this information with you. So what bioinformatics has done is, from 15 to 20 years, the drug development period has been reduced to something around 12 to 13 years. So you have reduced the time frame by about 5 years using bioinformatics. Secondly, the cost. I said that 300 to 800 million dollars cost was happening for drug development. But what bioinformatics has achieved is this drug development cost has come down to about $500 million on an average. So a 250 to $300 million cost reduction because of bioinformatics. Any problem, no? So now we come to what is bioinformatics. I have been saying bioinformatics all the time. So now I have to explain to you what bioinformatics is all about. So what is bioinformatics? Bioinformatics is basically a marriage between biology and computer science. So there is a husband and wife, biology and computer science basically. So it's a combination of two subjects. And you also have other subjects like physics, chemistry, mathematics, statistics, especially maths and statistics. Together it makes bioinformatics. So if you ask me to define bioinformatics in a very layman's term, I will explain bioinformatics as a very simple subject where you are trying to solve biological problems with the help of computer science and statistics. So the definition of bioinformatics is trying to solve biological problems using computer science, information technology, and statistics. So NCBI, the National Center for Biological Information, defines bioinformatics as a merger of biotechnology with information technology. So I will tell you why bioinformatics became very important. So all of you must have heard of the Human Genome Project. Have you heard of Human Genome Project before? The Human Genome Project. Anybody? Okay. So the Human Genome Project was started in the year 1989 and it was completed in the year 2003 in order to find out the entire sequence of the human genome. So the Human Genome Project found out that there are 3 billion bases of nucleotides in the human genome and there are 2 copies of the genome in every cell and there are about 7 trillion cells in the human body. So to get a rough idea of how big this is, I will tell you an example. I am a 32-year-old person here and if I start typing out the human genome today, A, C, T, G, 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 T, C, G, G, like that, if I keep on typing for the next 140 years one day, it will take me another 140 years to completely type out the 3.2 billion bases of the human genome, provided I'm typing at the rate of 6 nucleotides per minute without taking a break. If I keep on typing, it will take me 140 years to completely type out the human genome. And if you give me a printer and ask me to print out the entire sequence of the human genome, like A, C, T, G, it's all printed on paper. And if I have endless supply of paper and I'm printing and printing, if I stack the paper on top of each other, what will happen is the stack of papers will be taller than the Eiffel Tower in Paris. So tall will be the stack of papers because 3 billion bases is such a big number, 3 into 10 power 9 bases. And remember, what we are talking about here is just one cell of the human body, but there are 7 trillion cells on an average in the human body, and every cell in the human body has this genome inside. So if you take the entire DNA of a human being and make it like a rope, tie the DNA from each cell to the end of the other cell DNA, then you make a big rope. The rope distance will be as long as Earth to Pluto and back. So avlo length irukum. So human genome has so much information in it, and all the genome, the entire genome was sequenced by the year 2001 and totally completed by 2003. So At the end of the Human Genome Project, so much information was available. The scientists did not know what to do. So you might ask me, what is the big deal? I'll tell you. Today, on an average, a cell phone has about 32 GB memory, correct? All of you have a cell phone, and those cell phones will have 32 GB of memory in it. But back in 2003, 32 GB hard disk was not so common for even a computer. But when the Human Genome Project was completed, so much information had to be stored somewhere. It needed to be made into databases, and those databases needed to be publicly accessible so that everybody can access it. And the information needed to be made sense of, because all the genome is not coding regions. Only 24,000 genes are there, but 3 billion bases of DNA is there. You must remember that only 1.6% of the human genome is actually genes. The rest 98.5% is all junk DNA. We call it junk DNA because we don't know what to do with it. Maybe another 2-3% can code for RNAs, and rest of it is all intergenic regions and intronic regions, and these regions we don't know what to do with. So when all this information came into existence, humankind did not know what to do. Then they started the science of bioinformatics. But I would be misleading to tell you that bioinformatics started only in 2003. Bioinformatics has been there from 1970 onwards, but the actual bloom of bioinformatics happened only from 2003 onwards only. Even my department at Ramachandra University was started in the year 2004 after the Human Genome Project was completed. So much of information is there, and you need to make sense of that information. So now we come to how bioinformatics is helpful in drug designing. So for this, we have a particular science which is called as CAD. CAD means computer aided drug designing. Computer aided drug designing is a science which combines information technology, computer science, statistics, mathematics, chemistry, physics, and biology in order to solve the problem of drug designing. This is also called as computer aided rational drug designing process. So usually what we do is we identify a disease. Even in this case of corona, we knew that this disease was caused by a novel coronavirus, which was a mutant variety of the SARS virus, which caused the SARS epidemic in China and Southeast Asia in the year 2004. We identified the organism, and then we need to isolate the protein or some other.
Sir, your voice is not audible.
Sir, you muted your audio.
How long was I not audible, madam?
From last slide, sir.
Now I am audible, madam?
Yes, sir.
So even in the case of coronavirus, we know that corona is caused by novel mutant of the SARS virus. Am I audible now?
Yes, sir.
So it is caused by the SARS mutant, which caused the SARS virus was the one which caused the SARS epidemic in Southeast Asia in 2004. We know that the COVID-19 is actually a mutant version of that SARS virus. So we identify the microorganism, we identify the disease. Then we have to identify the protein or the target which can be targeted for the drug delivery. So here we know that we have proteins like spike protein, nucleocapsid protein, non-structural protein 1, non-structural protein 7, which can be exploited as a drug target because these are the important virulence factors of the virus, without which the virus cannot cause the disease. Or if you're going to target the spike or the nucleocapsid, actually the virus will die so that you can treat the disease very efficiently. So first you identify a disease, then you isolate a protein, then you have to find out a drug molecule which will work for it. So a lot of pharma companies today, ranging from Pfizer, Roche, and Novartis, everybody is working towards finding a molecule which can go and target this nucleocapsid or the spike protein and inhibit it. Even in your own department, there is research going on from plant compounds that can inhibit the spike protein. So world over, all the scientists, whoever is interested in virology, whoever is interested in drug designing, are all working towards finding a cure for the COVID disease. So once a drug molecule is identified, they will go for a preclinical testing with animals, and then they will go for a clinical trials where humans will be given the drug and adverse effects will be tested. So drug designing can be two types, especially computer-aided drug design. One is called structure-based drug design. Second one is called ligand-based drug design. So you might ask me, what is structure-based, what is ligand-based? So structure-based means you need to know the structure of the protein target you are going to send the drug to. So in this case, I was talking about nucleocapsid protein of the COVID-19 or the spike protein. So whether the structure is known, structure is definitely known. It has been deposited in this database called PDB. Have you heard of PDB? Protein Data Bank. If you have free time, go to this database called Protein Data Bank and type NCOV. NCOV means novel COVID. And you will get the structure of all the proteins in the COVID genome that have been elucidated so far. You can find them all. So how do you generally find the structure of a protein? You must either do a X-ray diffraction or you will do a NMR, then you will find the structure of the protein. And then you have the ligand-based drug design where the structure of the protein is not known, but sometimes you will know that this ligand will very well work for this disease. Say for example, Kiranelli Kashayam. You might have heard of Kiranelli, which is given to people who have hepatitis A or hepatitis B infection, and it has been used in traditional medicine in India for a long time. But only recently we were able to identify the exact phytochemicals which are responsible for giving the antiviral property to the Kiranelli. In that case, you choose the compound first and then you go for the drug designing process. So under structure-based drug designing, you have docking, then you have de novo drug designing, and you have virtual screening. In case of ligand-based drug designing, you have pharmacophore-based drug designing, then you have QSAR. I will explain what each of this is in very brief manner. So first one is called de novo drug design. So it is the iterative process. Iterative na repeated process. You have to do it again and again by a trial and error method. Then only you will know how to do it. So I'll tell you what de novo drug design is. Every protein has what is called the active site. Active site is the list of amino acids in that protein. Proteins are made of amino acids, correct? I guess all of you know that. So proteins are made of amino acids, but only few of the amino acids in the protein are going to be responsible for the drug to come and bind with the protein. Rest of the amino acids are only giving the protein as structural stability. So you identify the active site. Active site means the amino acids that are going to interact with the drug. So you identify the active site. The active site is going to be made of amino acids, and these amino acids will have some specific property. It might be aromatic, it might be basic amino acid, it can be aliphatic, it can be what do you call it, hydroxyl group amino acids, or it can be a sulfur containing amino acid like methionine or cysteine. It can be like that. So based on the active site, you will keep adding fragments of chemical entities, and you will synthesize a new drug molecule which will actually correctly fit into the active site. So some of the software that are used for de novo drug designing are Smog, Leapfrog, Builder, etc. One of the best software in the world is called Breed, B-R-R-E-E-D, Breed, which is made by this company called Schrodinger. Next, you have molecular docking. Molecular docking means trying to find out whether a drug will bind to the protein or not using computational methods. Initially, we used to have ELISA-like methods where you will find out whether a protein has affinity towards a drug-like molecule. Or they will go for a high throughput screening where the protein is immobilized on a tighter plate and drug molecules of different kinds are tightened out into the plate, and then you do a fluorescence spectrometry and find out whether binding affinity is there or not. But today we do molecular docking and do a virtual screening where you can screen more than 10 lakh compounds a day using a normal laptop and find out which molecule, but you can't find it out exactly, that I confide. Only 60 to 70% efficiency can be reached with molecular docking. But you can find out how much of energy is involved in this binding, whether this binding will happen efficiently or not, and how good the binding will be. So from about 10 lakh compounds, you can cull down it to like 1,000 compounds or 800 compounds. But from there, you have to do the wet lab experiments only. You have to do a cell line study. You have to do animal study. Then only you will find out whether the drug is good or not. There is no alternative for a proper wet lab study that you biologists do. But the bioinformatics is going to help you in cutting down the time frame. So this is exactly what docking is, trying to find the best fit between the drug and the protein. The tiger is the protein, Calvin, the boy, is the drug here. And there are two types of docking. One is called flexible docking. Another one is called rigid docking. Rigid docking is the olden day technology where we think that lock and key mechanism is correct. But today we know that lock and key mechanism is not perfect because induced fit model is the actual model that is explaining how an enzyme and a substrate work. Because when a drug is going to go and bind to a protein, the protein structure is going to change. The drug structure is also going to change. So you need to understand all that. So the best docking software today is called Glide, which is made by the company called Schrodinger. Then one of the popular software is called Autodoc and Autodoc Vina. And then you have a lot of free softwares like PatchDoc, Ligand FitDoc, OEDoc, ParDoc. ParDoc is an Indian product made by IIT Delhi. Now we come to ligand-based drug designing. Here, pharmacophore-based screening is the first method. Pharmacophore-based screening means, what it means is, you take any drug molecule, only certain parts of the drug molecule give it a drug-like feature. The rest of the drug molecule is all stability-giving features. So the three-dimensional representation of the features of a drug molecule which give it a drug-like property is called as the pharmacophore. So a pharmacophore of aspirin looks like this. So if you take the aspirin molecule, the sodium moiety is very important because it's a cation. Then the benzene ring, that's the aromatic ring. So that is also important for hydrophobic interactions. And also you have the salicylic acid group, the COOH, the carboxylic group. So that is also being a drug-like property. So important features of a drug molecule like hydrogen bond donors, hydrogen bond acceptors, cation, anion, aromatic rings, hydrophobic centers. Only these are considered, and a representation of the molecule in a pharmacophoric manner is created, and it is superimposed on the pharmacophoric feature of every other molecule on a database. So say for example, you have zinc database. Zinc database is a free database and it has about 15 lakh compounds. And you take all the compounds in the zinc database and you superimpose the pharmacophoric feature of your molecule against all the molecule on the zinc database, and you will find out which is the most closely occurring compound. So most popular software is Sparrow or Ligand Scout. Last one is QSAR. QSAR means quantitative structure activity relationship, which means that molecules which have a similar structure will inevitably have a similar function also. So what they do is they predict the physiochemical properties of the molecule and try to create a regression model trying to find the mathematical association between the physiochemical properties with the biological properties. Say, for example, the inhibitory concentration or the enzyme kinetic concentration, etc. So there is something called, I'm not going into the depth of it. If you want, we can have a separate class on docking, separate class on de novo drug design. Everything is possible. So there is something called the Lipinski's rule of 5. Lipinski rule of 5 is basically a rule for oral bioabsorption. If you are going to give any drug orally to a patient, then it must follow these rules. The molecular weight must be less than 500. The water octanol partition coefficient, which is called the log P, must be less than 5. The number of hydrogen bond donors must be less than 5. And the number of hydrogen bond acceptors must be less than 10. Only then a drug will be a drug which can be given orally. Else it cannot be a drug molecule. This is like a filtering that you can do for any molecule. Say, for example, you are doing some work on pepper or chili or karvapala, whatever. You identify some new compound and you want to see whether it will have a drug likeliness or not, whether it is likely to be a drug or not. Then you can check out these filters and see whether it's obeying the filter. If it's obeying the filter, then there is a good likelihood that it's a drug. If it is not obeying these rules, then it means that molecule cannot be given as a drug. So some of the software are ChemProp, Mall Inspiration. The free software is SwissADME, which is very easy to use. So I'll wind up with this. So if you have any questions, you're free to ask me. And if you have more sessions in future, you can always contact me. I'll leave my phone number also. So if you have any doubts, you can WhatsApp me for your research work or something like that. Thank you. Madam?
Yes, sir.
Any questions, madam?
Sir, one question
Yes, madam.
from Gayatri Devi. She has asked how to detect active site of target of protein. How to detect the active site of target protein to perform molecular docking.
Okay. Okay. So you can go to the software called Computer Atlas of Surface Topography of Protein, CASP. Are you able to see it, madam? So you can go to the software and you can put the PDB file for which you want to find the structure, the active site. So you can just add your protein here and it will find, you can just type the PDB ID here, or you can add your own protein and it will give you the list of active site residues. Like for this protein, the active site is in this red color region and the list of amino acids is given over here. So you can use this software. This is like one of the best software. So you can upload your PDB file here and you run it. Hope this answers your question.
Sir, another person, Arivoli, has asked, what is the effect of bioinformatics in research field?
You mean in terms of job opportunities or in terms of utility?
I think she means in terms of utility.
So as I told you, you can do fast screening of molecules. So in 1990s, you can screen only about 1,000 compounds in one day. And that will also cost several lakhs of money per day for pharma companies. But today, if you have a library of 10 lakh potential compounds that can bind to a particular drug, particular protein target, you can screen 10 lakh compounds in one day and cut the list to about, you can filter it down to about 800 or 900 compounds. From there, you have to do your wet lab study again, but the time and the cost is being reduced drastically by bioinformatics. Another area where bioinformatics is very useful is next generation sequencing. So next generation sequencing is an alternative for the traditional Sanger method, which is using parallel sequencing technology. So back in 1988, when they started the Human Genome Project and completed in 2003, sequencing one single human genome costed about $7 billion. That is more than the worth of the Infosys company. So 1% total genome sequencing, $7 billion. But today, if I want to get my sequencing done for my entire genome, it will cost me less than $10,000. So $10,000 is approximately 7 lakh rupees, which might seem expensive, but it is at least affordable to at least the richest of people. But back in 2003, it was $7 billion. So compare the cost. So this is only because of bioinformatics, because so many algorithms have come which are capable of doing parallel processing of data, and assembly and quantic mapping are done so fast that bioinformatics has become a boon for drug development. Any other question, madam?
Participants, if you have any questions, please put in the chat box.
And for the first person who asked the question about the active site prediction, the best method is actually, there is a database called PDB-SUM, which is the sister database of the protein data bank. So what you can do is, if the protein structure is already known in PDB, you can just put the PDB ID here. Say, for example, 1BOT is one PDB structure, and it will show you the ligand interaction map of that molecule. So the ligand here is sulfuric acid in this case, and you can get the list of all the active site amino acids by clicking on this. Are you able to see? So you know that tryptophan 84, glycine 118, serine 122, these are the amino acids that are going to interact with acetylcholinesterase based on this. So if the protein is not there on PDB, use Cas-P. If the protein is there on PDB, then you can use PDB-SUM. Any other question?
Hello.
So there is a question from Ayani Swami. Is any target protein identified in COVID-19 using bioinformatics? Can you throw some light on that?
Repeat, madam, sorry.
Any target proteins identified in COVID-19 using bioinformatics? Can you throw some light on that?
One of the important target protein is spike. SPIK is spike of, I'll show you the protein, that will be very helpful for you. So go to this PDB database. Are you able to see my screen? So here you type NCOV, novel coronavirus. So here all the proteins in the coronavirus are shown. So this is the main protease of the coronavirus. This is one of the main targets. So a lot of people are working on blocking this particular protein in order to cure the disease. So the idea of this protein is 6xB1. Or you can also target the nucleic acid or the spike protein also. Any other question, madam? but most of the work is on the protease or the spike protein.
Sure, one more question, sir.
Yes, madam.
Hello. Is there any one violation in Lipin case rule the drug is acceptable? If there,
I got the question, ma'am.
yes.
So Lipin's case rule is only for oral bioavailability. So the drug need not be given orally all the time. Sometimes the drug can be given intravenously, or it can be intravenously, or it can be given subcutaneously or intramuscularly. There are multiple ways of delivering the drug into the body. So if the Lipinski rule are not followed, then it means the drug will not be a orally bioavailable drug 90% of the time. There is still a 10% chance that the molecule can be orally bioavailable. But the better alternative will be to administer the drug by some other method, say for example like a subcutaneous method or intramuscular method that can work really well. You cannot swallow it as a tablet, but it can be given as injection.
So one other question from Palaniswamy. Up
Okay.
to what level of energy during docking study is considered a good lead?
That depends on the software you're using. So if you're using Autodoc, I guess that would be the software you're using. More than -5 kilocalorie per joule would be a good binding energy. So the energy here we are talking about is the what is called the Gibbs free energy. So are you able to see my screen? So Gibbs free energy is called del G. So biochemically del G equal to del H minus T del S. That means the change in entropy minus the temperature and the change in entropy. So this del G must always be a negative value for a spontaneous reaction. It must be less than 0. Only then the reaction will happen. So if the del G is a positive value, then that your docking is not feasible at all. I mean, the drug will not bind to the protein at all. So your value must be less than 0 for sure. If it's more, if it's better than -5, then it's good. Sorry, not less than, greater than. So it should be greater than -5.
Okay, sir. Another question.
Yes, madam.
Can you kindly suggest the parameters that has to be checked to publish docking result in journal except binding energy?
It's always good to find MMGBS. So it's always good to find the MMGBSA value. That is like validation of your docking. MMGBSA means molecular mechanics, general bond surface area prediction. So if that value is also good, then that means your docking is very good. Or other alternative is to do a MD simulation, molecular dynamics simulation. But you will need a high level of computational power to do that. But you can collaborate with some institution like Madras University, especially Dr. Gunasekharan's lab. So you can contact them, get a collaboration, and go for it.
Okay, sir. Thank you, sir.
I was seeing somebody asked about album arsenic. I am honestly not a believer of homeopathy medicine, so I wouldn't comment about it much. So I will not believe in homeopathy much, because homeopathy is based on a particular property called water memory, which has not been physically validated anywhere. So I am not a supporter of this, so I will not comment much on that. But I don't think there is any commonality between cholera and COVID, because the basic pathogenesis of cholera is based on the influx of chloride ion leading to a dysentery and rice water stools. I don't think COVID works like that. The pathogenesis of COVID is absolutely different. So, as a molecular biologist and a bioinformatician, I don't see any similarity between cholera and COVID, except for the fact that both are very serious problems. Cholera we have tackled to a large degree. We have a lot of medication against cholera. COVID, we don't have any such solutions. But other than the fact that both are very deadly pathogens, there is no other commonality.
Thank you, sir, for your valuable speech, and it was more informative. I
Thank you,
invite
madam.
Dr. Meenakshi ma'am to propose vote of thanks.
Respected,
Meenakshi ma'am.
yes, respected speaker of this evening, Dr. Abhinan. So you have given the role of bioinformatics in the drug discovery in a simple and an easy language so that everyone can understand. And you have also clarified all the doubts the participant has posted on. And the application of bioinformatics has reduced the time for developing a drug and putting it into the market, as well as it has also reduced the cost. So I think the students would have been beneficiaries on attending this lecture. Thank you so much, sir, for
Thank you,
accepting our
madam.
invitation. Thank you.
It was a proud privilege to be here, madam. Thank you. Thanks a lot.
Welcome.
Participants are requested to stay online for the next session. I kindly call Dr. Vijayalakshmi ma'am to address the resource person for the second session.
Good evening to one and all assembled here. It's my great privilege to introduce our guest speaker, Mr. Bala Bhaskar. Mr. Bala Bhaskar has 19 years of teaching experience. He started his career as an assistant professor at SRM Arts and Science College. He has done a lot of improvisation and improvement. He has acted as the coordinator of IQAC cell. He has been the editor and founder of his department newsletter, and he has also been the coordinator for the examination wing. Later on, during his career, he had a passion for industry and he moved on to diagnostic industry. In the diagnostic industry, he was acting as a leader or the head at Vimta Labs, Hyderabad, for more than a year. Then he moved on to his present position, where he is now the product manager at PrimeGen Lab Private Limited. He has a craze to learn a number of online courses, and his biodata shows that he has learned a number of online certification courses. A few to name are NPTEL and other bioinformatics courses. He has published more than 15 papers to his credit, and he has attended a number of workshops. So on behalf of the Department of Biochemistry, I warmly welcome Mr. Bala Bhaskar to deliver his lecture on diagnostic sector for our students. Over to Mr. Bala Bhaskar.
Sir, you you're not audible, sir. Sir, kindly unmute your audio, sir. Sir, please unmute your
Hello.
audio, sir.
Yeah,
Yes,
hello.
yes, you're audible, sir.
Yeah. Good evening to all of you. Thank you very much for the organizers and the principal of Bharati Women's College for giving me this opportunity to present on this topic. Are you able to see my screen, everybody? Hello?
Yes, sir. Yes, we can see the screen, sir.
Yes, able to see my
Yes,
laptop
yes.
screen?
Yeah, yeah.
Is it coming up, madam?
Yes, sir.
Yes, sir.
Okay. So I think the previous session was more on bioinformatics and was giving a very clear-cut idea on how we would be able to have a career with bioinformatics and how we can actually present on do more work with respect to bioinformatics. So when you're looking for a life science student, most of the time when we are studying and when we are going across our first year to second year to third year, what we would be focusing on as our next future is after you finish your BSc, you would move on to your MSc, and then after MSc, we would think on research, and after research, we would be thinking on some scientist labs like that. We will be just moving on and on and on. But there are going to be lots and lots of other career options also for life science graduates, more than what we are actually thinking. So before we get on to this actual topic, let me just give you a small story, which was somewhere around 1995, 1999, when people were choosing a lot of computer science streams, and they were thinking on how we are going to get a job after we finish our graduation, dig engineering, etc. But come at the Y2K problem, and suddenly you found that there have been thousands and thousands and still thousands of people who are actually looking in to complete their degree in BE computer science or BSc computer science and land up with the IT job, and they keep working on. So that's how the computer science field bloomed, basically. Like that, now with this present situation of corona and testing and how many different types of testings are being required, we from the diagnostic field right now, we just feel that this is the time for the life science graduates for the next 2 decades or so. Definitely we are going to have a bloom. And so that's why I chose this topic when I was given an option to present. So I just chose this topic of giving scope of life science graduates in healthcare industries. So we'll just move on. Let me have, and thank you very much, ma'am, for giving me such a wonderful introduction. I'm really honored and humbled by that introduction. And I'll just give you a small intro on myself. I'm Balavaskar, Product Manager of PrimeGen Healthcare Laboratory Private Limited, Nangaluru, Chennai. We'll get in with our company a bit more later. So my first slide, when you talk about healthcare industry, so what is going to be the healthcare industry? Now, healthcare industry has become a part and parcel of our everyday life, and it has become one of the largest industries in India in terms of revenues. So normally healthcare and revenues, we don't associate too much. But nowadays, this healthcare industry, not just as a revenue producing option, from the career and job perspectives, also the healthcare domain is becoming one of the most important and both money spinning sector to work in. So the first myth that I have to break with all these graduates, young graduates who are listening to me, I presume from the participants list that most of them are going to be students from across the different colleges, co-ed and women's college. The first myth that you have to break on is lab industries or healthcare industries is not only for one particular gender. It is going to be for both of you, provided you're going to be very smart and very choosy on what you're going to do. And it is going to be like right from the beginning of a lab till you perform and till you give back the report or till you give back your research products or whatever it is going to be, every place it requires specialists. It is like it is just not like anybody can do any type of job or anybody can just come and learn and do it. You have to be a specialist in that particular domain. Only then you would be able to succeed. So that's going to be the case. Career and job perspectives are aplenty in this healthcare domain sectors. So it is just going to be, you need to be pretty smart and proactive to see what's happening around you to choose and move on in this healthcare industry. So when you talk about the healthcare industries, since we are in the laboratory diagnostics, I would be focusing more on the healthcare with respect to laboratory diagnostics, and wherein we talk about three major segments in healthcare and medicine. There are three types. One is going to be the preventive medicine, the other one is therapeutics, and the other one is diagnostics. So why I'm giving all these details is going to be because you need to understand in which are the areas that you might get in once you finish your basic education, whether it go to be a BSc or a MSc or a PhD. You should be having an idea of where you would be able to get in and how you have to equip yourself. So for that, you will know that there are going to be three different types: preventive medicine, therapeutics, and diagnostics. So what is preventive medicine? Preventive medicine is going to be involving routine checkups. So preventive, as the name itself suggests, that it is going to be something which you're going to do on a basis where you're going to just ensure that you're going to be in a good health. So that's what I say as by clinical definition. It involves routine checkups, immunization, or screening when an individual is symptom-free and healthy. So for example, beyond after a particular age, you just go have a blood glucose test or you do a cholesterol check. Those are all categories that come under preventive medicine. So what exactly preventive medicine helps you out? It gives you and your doctor a snapshot of your current health, so that what might happen to you across or over a period of time, or what might affect you. For example, if you are going to have a familial heredity of diseases, then the preventive medicine becomes an important one, so that you will have a constant check on what you're going to do. And therapeutics, this is going to be one where you're going to, the subject is going to have a disease or a treatment, and they are going to have treatment of diseases or disorders by medicinal drugs or methods. So that is what therapeutics is. And finally, you have the diagnostics, which is going to be the most important one, where the individual undergoes all the symptoms or risk factors, so they go for a testing. So it involves tests or procedures that are being ordered by a physician, and then you have to go to a lab, give your sample, and then you follow up on what is that. So the diagnostic care is basically the follow-up of an issue from an irregular test result or an existing illness, injury, or health problem, or already being treated by your doctor. So this is what, in simple terms, we just put it like a lab, a general lab, that whatever a lab that is nearby to your place, or a bigger lab. Those labs come under the broad category of diagnostics and preventive to such an extent. And when the same thing is going to happen inside a hospital and a hospital associated lab, then it becomes therapeutics. Okay, am I going fast? Hello?
No, sir.
Yeah, okay. When you talk of diagnostics, this diagnostics can be of two different types, like how we always study in science as in vivo and in vitro studies. You're going to have two different type of diagnostics, primarily, which is called as in vivo diagnostics and in vitro diagnostics. So when you're going to go in with a study with respect to bodily fluids, then it becomes to the category of in vitro diagnostics. And when you're going to have your imaging studies, then they become your in vivo diagnostics. So in vivo diagnostics require more of specialization with respect to, you can see there, You have MRI, magnetic resonance imaging, ultrasound scanning, sonography, X-rays, mammographies, positron emission topography, PET scans, colonoscopy, CT scans. These all come under in vivo diagnostics. So they are going to be directly performed on a human body. So this is called as in vivo. And then when you talk about in vitro diagnostics, this in vitro diagnostics, they involve processing and investigation of biological samples collected directly from the patient, such as blood, urine, saliva, tissues, feces, etc. So these human samples are analyzed in a laboratory setting to detect and quantify the presence of a specific biological marker. So I hope you understand what is a biological marker. Being science students, you will know there are going to be certain markers, certain molecules which will be present at elevated levels during a particular disease conditions. So when you do a testing and you find a particular marker, say for example a cancer antigen, then if that level is going to be high, then you would be able to have an interpretation that, okay, the sample, the subject, the person is going to be having some symptom of a cancer, and so they have to go on for the treatments necessary for a cancer. So the biomarker could be a circulating protein, pathogen, electrolyte, or even a genetic mutation. So so that is going to be on the type of diagnostic. So now what is the diagnostic market in India? So this also we should know, because we always talk as an academician. I think madam, when she was telling me, when she was introducing about me, she had told me, she was telling that I'm having close to 15 years of teaching. And I discuss with most of the students, I tell them that, see, we have a bright future after you finish your graduation. You would be landing up here, you would be landing up there, you can become an academician, or you can become a researcher. Academician and researchers are ever living platforms. As long as you have an opportunity to work, you'll always get in. But this diagnostics, again, this is a new platform for lots of us. And a lot of the people, you don't have an idea of how this diagnostic market is blooming. So this slide basically tells you how exactly this diagnostic market is in India. And you can find that the in vitro market is going to be more larger than the in vivo market. So testing labs, you have lots of labs, lots and lots of labs around. And you can see that every lab is going to come with some sort of a specialization, and they ensure that they are going to be the best in that field, so that the testing and the quality as well as interpretations are going to be at the top notch. So you can see here, as per the market research report, you can find here the biochemistry instruments and reagents itself. They are going to have a market of around 1,742 crores, which is a data which we had collected in 2018. And you can find that the in vitro diagnostic market by default, it's coming close to 6,300 crores with 2018. No wonder now it would be somewhere around, no wonder it would be somewhere more than around it would have doubled by this time, seeing on the market that is going to be there. And you can also see that 70% of the diagnostic market is being dominated by the in vitro segment. So the need for life science graduates, more specifically biochemists, microbiologists, and biotechnologists, are always in plenty. But the only drawback that we have is we don't know how to tap on it. And when you talk about the in vitro segment itself, the measurement of the biomarker is only by two types, either by a standardized laboratory developed test or an in vitro diagnostic test. So what do you mean by a standardized laboratory developed test? Is going to be a test that was developed by within the lab itself. So we prepare a method and we test the method, we get the approval. Then that becomes a standardized laboratory developed test. Or an in vitro diagnostic kit, a device or kit, that is going to be a kit that is previously available. You purchase that, you you purchase that, and you
Sorry for the interruption. Bala sir, your mic is muted.
Bala, your mic is muted. Bala,
Sir, kindly unmute, sir.
yes, is it audible now?
Yes, sir.
Yeah. So may I know where we stopped basically?
Last
Where
slide, sir.
last slide, okay. Yeah. So this is the Indian biotechnology industry's value, which is going to be around $11.6 billion. And you can see there by 2025, we're going to hit a 100 billion mark. So these are all the broader values that basically, which has been talked by the bigger companies, governments, countries. But the catch that you have to understand from this is to get into this particular volume of investment and money, how much of labs should come in and how much of quality labs should come in. So quality labs, they're just not built, but they require quality people, and quality people refer to only to all of you budding young students who are pursuing life science, who should have the interest of exploring into this market, and you will just not just benefit for the country or for the market by itself, but you will also be greatly benefited. So the recent spike in this pace of growth can be on. Okay, what is the reason for this? You can have a lot of things being attributed. And you can also be listening to this word, which is called as medical tourism. So tourism, this is something which has come up in the latest word, which we call as the medical tourism, where India has become one of the hub for a lot of countries because of our technological innovations and knowledgeable doctors, professionals, and healthcare people. More and more people from Southeast Asia and a lot of other countries, Middle East countries, they want to come and have their treatment in India. So that is why we say that we have become one of the leading destinations for medical tourism. And this is a stat that I'm presenting to you, that we are going to reach as much as 1.6 trillion US dollars over the next 4 years. So this broader picture also gives you an idea of how much the field is going to grow in the coming years. So you have to equip yourself, and you should be a very broad-minded, not to confine your opportunities only to, okay, I can be doing only this. I keep repeating this again and again because we always have a myth that beyond this point we will not be able to do, or beyond this point we will not be able to work. See, this 2 months of Corona has literally turned the world upside down. People who were telling that they were not able to work from home have started to work from home. People who said they don't know how to operate a phone have started to operate a phone. Like that, you are going to have so many options. The only thing is you have to tap on the right option. And I'm giving you one more piece of information for you. The biochemistry portion is going to be 38%, the major share. So which means biochemistry, it's going to be spread across all the life sciences, whether you're going to be a microbiologist or a biotechnologist or the biochemist by default. Fundamentals of biochemistry always gives you that plus point that you can shine anywhere in the diagnostic market. Recently, I saw one of a person who was in the marketing industry of a healthcare company. He was telling me very happily that, sir, whatever I had learned in biochemistry has helped me to promote my product. So you should not confine yourself like, okay, I'm always just going to wear a lab coat and hold a pipette and do things like that. That's good. That's your passion. You can do it. But if you're not able to do it, you have got plenty of options. Don't just let out, or don't just change your field just like that. Every field has got its own throw and its own share of openings. The only thing that is that you have to catch on to it. So you can see here, biochemistry is holding 38% of the shares, or 38% of inputs are going to be from biochemistry. And then you have it in immunology, hematology, and the other things are following. So in vitro diagnostic market, you can see here there are enough career and job opportunities. So some things which we keep telling you again and again, you will start believing. This is not something which we are going to tell you as an advertisement or saying that, see, you study this, you're going to get a good job or a good career. You put your hard work and you be smart to look out in the correct options and the correct openings. You can see how much of opportunities that you're going to be there. In the coming slides, I will present to you what are all the different responsibilities that you can take inside a lab itself. So now, why is it making it easy? Why the fundamental theoretical background of a life science is going to help you out? You can see here the impact of automation. Every country, we don't do any more things in manually. Everything has become automated. But automated also needs a person to handle it. A computer, an algorithm, you devise an algorithm, you can say that this is going to help you to solve a problem in seconds. But who devised the algorithm? A human being. Our human genome project, the softwares, the biotechnology softwares, the bioinformatics softwares, who is designing it? A human being. But the automation, the final product is going to be time consuming, time saving, precise, and perfect. So the impact of automation on diagnostic jobs, you can see here, there is going to be both the positive and the negative side. Again, there is no blind truth. Negative impact is there because if you are not skilled, if you are not equipped, then you will not fit inside the job. So, and you cannot get yourself equipped, you cannot get yourself skilled after 10 years or after 5 years. So when you are a student, when you are just getting in to start your career, you should be in a position where you will not be able to know everything, but you will be knowing to say that I have the confidence to get in. I know an idea of this. So you can see here, there is going to be high job growth if coupled with technology, diagnostic domains. They can going to create a lot of job opportunities. So you can see here, as of now, you maybe in classes you would have talked about in a laboratory, we do in our labs, we do on manual lab testing. But you can see here there are semi-automated analyzers, automated analyzers, and we have basically we have just right now we have for simple blood tests, genetic screenings, and all these things have become automated. But sooner or later, you're going to find complicated tests also, they're going to get automated. So why the job opportunities are going to become high? Again, we are going to be the reason. The lifestyles, hectic schedules, stress, and complex lifestyles, they are going to bring in some sort of a physical change, a complication to us. So we are going to go for testing and testing. So this is going to be, like we will wish the person is going to be healthy. But because of the schedules, the stress, and the lifestyles, the person gets some sort of a complication at during some stage of his life, and testing becomes inevitable. And then prevalence of severe diseases, again, this is going to be correlating with your lifestyles, diabetes, hypertension, cardiovascular disorders, infectious diseases across the population. So the need for testing, trust me, from now onwards, for everything, for every passage of every 5 years, 10 years, the need for testing is going to improve. Who would have thought of this COVID testing some 5 years back? Why 5 years back? Last year, would we have ever thought of this COVID testing? Now people are not bothered about LFTs or RFTs. They are telling that we need to have a COVID testing done. How do you do it? You do by a RT-PCR. You do by you're doing qualitative. You're doing quantitative. People are so curious to know about it. So diseases are not going to decrease, but they are only going to increase. Maybe they are going to be known, or maybe they would be found out by researchers like you, saying that this is going to be a new one. And then increasing awareness among the educated people. So this is a very important point. It is not that only people who are suffering should go and test themselves. After a particular age, we say that after 40 or after 45, it's good you have a annual complete health checkup. You just test yourself. It is good. So that itself is becoming another factor for diagnostic sector to thrive on. And then limited presence of organized players across the country. So this is where we talk of quality and specializations. Not every lab can afford specializations. There can only very few labs which can afford all the specializations or quality. Quality means you need to have an accreditation, like how we talk of ISI and ISO. Labs, they're going to have an accreditation from NABL or ICMR, like that. So not every lab can afford for that. And it is no easy process also. And then the last three, where we talk of the healthcare IT industry. So IT industry and healthcare, they are going to be, this is where we talk of telemedicines, then we have your medical transcriptions, and then you have insurance, IT industry, insurance processes, and all these things. Again, this also gives you a demand. So for growing number of diagnostic jobs in our country. So now let's get into the business. The top diagnostic companies. I know that you would be able to list out quite a few, but these are some of the top diagnostic companies that we have in India. SRL, then you have Lalpath, then you have Abbott, then you have Quest Diagnostics, you have Thyrocare, and I'm sure that you would have heard of Medal. I have not listed everything, but these are some things that will commonly that comes off whenever we talk of top diagnostic companies in India. Even if you search a Google search or you go on anything, you will find that these are labs that have been listed. So how come they have been given a top diagnostic company? What is the criteria for that? The criteria for that is going to be only quality, quality, quality. No compromise on quality. And of course, the next remaining factors come on how much of automation they have, how quickly they can give you the results, how reliable their results are, what is their network spread across the country, like that, and so on and so forth. But the most important thing is quality, like how we rank up students, the toppers, the second, then the middle, the average, like that. Diagnostic companies also are going to be pure and purely based only on their quality. So now when you talk of education options for a career in diagnostics, now what are the different special?
Sir, you're muted, sir. So please unmute your audio, sir.
Yeah, I'm unmuted now. Audible?
Yes, sir.
Again, just move on. So when you talk of what are all the major disciplines that you need to study, you can see here I have listed all of them almost, but you can see biochemistry, microbiology, molecular biology, genetics, and clinical chemistry, diagnostics. They are going to be the best career options when you talk about in terms of diagnostics. Pharmacy followed by specialization in analytical chemistry or diagnostics. We will leave the last one, which is going to be for MBBS. So again, what is the average salary that you can think on? So this is again, sir, you're talking so much about diagnostics, you're telling there are so many things, but what could be the salary? So it's like the more you spend time, the more you ripe, the more you get experienced, you will be most sought after person by the companies and by the industries. So you can see here, I've given you the entry level. You can see how much you can earn. You can earn up to 3 lakh per annum. That's going to be the beginning one. And then the mid-cap professionals, your maximum scale would be around. My slides are visible for all of you?
Yes, sir.
Yes, sir. Yes, sir. Yes.
Okay. So the mid-cap professionals, they are going to earn somewhere around 7.5 lakhs, and the senior professionals, you can see that that's an whooping amount for you per annum. So it's again, you can't expect it overnight. You have to be in the field, learn the field, understand the nuances, grow with the field, grow with the industry, and I'm sure that you would be having a great future in this particular diagnostic field. So now when you talk of labs, so now having discussed on diagnostics and what you're talking, like what is the market and all those things. So how are the labs? How can the labs be defined or categorized into? You can have the labs being categorized in 3 types based on ownership, function, and specialization. So ownership becomes the government labs and the private labs. So for example, I'm working in a private lab and mine is a private lab. And then the second category of lab is going to be based on function, where you're going to have general or specific diagnostics. So again, we can say that basically our lab, we work on prenatal screening. So we are more of a genetic testing laboratory. So with that not all the labs can afford to or can have. So those are called as general labs, and we become a specific diagnostic lab. And then you have based on specialization, where you talk of clinical labs, blood banks, molecular biology labs, or public health labs. And this is where I said, sir, you've been talking of diagnostic field. What is my role here? You can see these are the different designations. The designations are looking so catchy, right? From a phlebotomist, which is as simple as going to be but the most important role in a lab, from simple blood collection, from a phlebotomist to a microbiologist to a biochemist to a histotechnologist who does work on my histopathology department, then cytogenetic technologist who does on chromosomal analysis and genetic analysis, pathology assistants, medical lab technicians, medical technologists. So these are all going to be lab related works. And then you have somebody called as a quality manager who will supervise the lab, who will ensure that your lab is going to be matching, complying with the standards specified. These called as a quality manager. Then you have got something called as a technical supervisor, a person who ensures that the machines, the kits are going to be perfect. They are not going to be outdated. The current one is there and it has been validated. It is all giving quality results. All those things are being handled by the technical supervisor. And finally, the top notch, where we talk of the lab supervisors and the lab directors. So just imagine yourself, your name, whatever, how you can just call it as, and you just add one of the designation, and you can see that In the beginning, it might look very simple for you. Oh, am I a biochemist? But then you know that in a biochemist, 3 years down, you've been called as a senior biochemist. 5 years down, then you become a much more important role. 7 years down, you would be called as a department in charge. 10 years down, you become a technical head. And then who knows, if you start your own lab, then you will become a lab director. Of course, the qualifications have to be filled on. The eligibilities, all those things will be changing and you have to fill on. But you can see here, to begin with and to the top, there is going to be a continuous growth process. So there is absolutely nothing there where you can see that you're going to have a lag, or you can say that my career is getting stunted at this growth. I will not be able to move forward. There is going to be a movement. If you're going to be aware of what's happening, you keep on learning. Like all teachers say, you have to keep on learning. It is not that once you settle into a job, you say, okay, my role is only this. You keep on learning, you master that, and then you have to move to the next level. Only then, whether it is going to be a private organization or a government organization, will they be inclined to take you to the next level. So it's always 80% from your side, and the remaining would be from the other side. So these are the different roles in laboratory that you can give. And so I think most of you would now know, or just start thinking that, okay, so I have got something or the other. If I finish my graduation and leaving, I'm not touching academics and research, though I can very well talk about it for hours together. I've been doing that for hours together. But right now we are shifting and looking in only in respect of diagnostics. So I'm limiting my talk only to this. So what I expect in a lab in terms of workspace. So now, okay, you want to work in a lab. What will I look into a lab? What have to be looking into? As a student, you listen for 5 hours to five different lectures. You do a practicals like that. What would be your work style? What is required from you? You have to be determined and focused, and your skill developing becomes a prerequisite. If you don't know, learn it. If you know, master it. There is no other choice. There is no other excuse behind that. And be a team player. This is going to be one of the most important attributes. You have to gel in with whatever team that is being provided to you, or you're going to join in. Understand the strengths, understand the weaknesses, go with the team, learn with the team, and only then you would be recognized. And you should know what is the organization of your particular laboratory. It might be a very simple lab. It might just have 4 people. But then you have to report to one person. That person will be reporting to another person. That person is going to be the person who is in charge of releasing the reports, who will be handling clients or who will be handling patients. So you should understand. You should not, if somebody asks you, I want to come and have a test in your lab, whom should I ask? You cannot say, no, I am just only a biochemist, or I'm just only playing this much role in this lab. I don't know about that. Then they will say, Arey, this person does not know anything about his lab. So why should I come and why should I go to his lab? I will go to a better lab. So you should be understanding the organization. You should know what your team is. You should know what your lab is, how good you are. And then this is again, long hours, demanding work style. Both of them go hand in hand. See, some pandemic situation suddenly come, your workload increases, a situation that crisis comes, machine fails, testing fails, you stay back, you work. That's an emergency. But when you go in for a bigger labs, you would be working, trust me, you would be working 12 to 14 hours, minimum 10 hours. And the work side would be pretty sapping. It would be a bit demanding. You would be doing the same work again and again till you move on to the next department.
Sir, you muted your.
So you will be having demanding work style, and you should have patience. You have to take out how you are handling the first sample, the same concentration, same focus, same determination, and the same skill has to be done to the last sample which you are handling also. So you need to have these 3 are going to be very, very, very important when you talk of it. Long working hours, demanding work style, and a lot of patience. So now why am I telling you this? Because you are just going to start in. I hope that there are going to be a lot of students as well as some of the academicians in there. You can tell your students that when you get into a lab, when you get into a workplace, This is how you have to be. So you have to get mentally prepared for that. And that is why I'm telling you this also, wherein you need to have lot of work hours, demanding work style, and a lot of patience. So next is how to apply. So you've finished your job. Now you're going to get in. You have to check in for the labs. You have to enroll your resume online, and you keep your options open. If you're going to be a biochemist, if you're going to be a microbiologist, don't just look in for microbiologist or biochemist. Across the life science, whatever options are there, apply for it. If they feel that you are fitting their requirement, they will call you for an interview, and from there you can take it on. You just don't narrow down your options. You please apply to every option that is relevant to you, and then you see how it is working on from there. You take it from there, and if they find that you are eligible for a particular posting, they will call you for an interview, or they might even provide you a training, and from there it is going to be taking up. So prepare a solid resume. As a student, how could your resume be built upon? All you can say is, sir, 3 years or MSc, MPhil, or a PhD. What more I can add? I can just keep adding on that. That's where your vacation comes into play. That's where your skill development comes into play, which I'll tell you in very short time. And then how to prepare for an interview stuff. The technical stuff, the subject knowledge, you always do it. But that is alone not is going to fetch you a job just like that. Because right now you will understand that there are going to be 3 different levels. The first level itself is going to be a HR level, and then you have the technical level, and finally you will have the management interview. So any place when you're looking to a bigger labs, bigger organizations, these would be the 3 different levels that you have to cross on. So first, get to know about the company where you're going to apply. What are they specializing in? So for example, if you're going to take an ABC lab, browse through the lab, see what are they, see where they are having, where is their headquarters, or if they are having any branches, how much they are doing, what is their profile, what is their logo, what is their brand. You might be able to share them. Even the HR is going to ask you like, how did you know about this? How did you know about our company? You can say that, I know that you've been here for so long years, and I know that you're specialized in this. So I want to join, learn, and grow, which gives them an input, a positive input, like, okay, the candidate has done a small sort of an investigation about the organization itself. That means he's interested. So this is the main important thing, apart from what your educational stuff, a technical knowledge. You have to know about the company, understand what they are specializing in, and then know about the different departments. Again, what made you apply to this? Basically, I'm a graduate in biochemistry, or a biotechnologist, or a microbiologist. I know that you have a department from your company profile. I saw that you have a department there. So I want to join the department and I want to learn. So that's how it's more of communication. It's more of conversation. It's more of actually impressing and giving the feel that you are interested to get in with the company. That is what will allow you to clear the first round. There you say that I know how to handle a GCMS, I know how to run a HPTLC. They will just come back to again this only. So know your rounds. If it is going to be a HR round, you have to talk accordingly. If it is going to be a technical round, there you cannot say that I know that you have got 5 departments. There you have to put your technical stuff. I'm very good in doing this. I'm very good in extracting. I'm very good in HPLC. I'm very good in this particular area. So I would be able to support your existing team. Like that you can talk on. So each and every round is going to be, so gone are the days where, you just go with some sort of a reference or some sort of a recommendation or some sort of even your academic marks. Just by that, they are not going to take you in. They are going to see how competent you are. They are going to see whether you would be able to sustain the field, compete with the present scenario. So for that, you should be developing all these things. Maybe that is what we teach you in this soft skill, personality development, how to approach interviews and all those things. They are no time passes. Remember, if you are learning those skills properly, they will definitely help you to get through the first round so easily. You'll be so confident presenting to them, talking to them. Technical knowledge, again. So this is going to be depending on the type of industry that you're going to choose. Basically, clinical biochemistry, analytical biochemistry, molecular biology, biotechnology, and biostatistics. These are very, very, very important. If you are able to have a know-how of all these areas, you can very well get an entry into the diagnostics and you can grow into it. And finally, you have the quality assurance, which is what we talk on quality managers and technical managers, wherein once you get into a lab, You have got a lot of testings, accreditations, wherein you have to maintain documentations, and you have to simultaneously look in whether the lab is doing properly and at the same time file your records. That is what becomes quality assurance. But as students, these are some topics that you are more familiar with: clinical biochemistry, analytical biochemistry, molecular biology, biotechnology, biostatistics. I remember in an interview wherein one person had come and he said that he is good in clinical biochemistry. The next question that was thrown to him in a technical round was, so what do you think is the normal level of electrolytes in a for a human being or for a person? He was not able to answer. And then they asked, what are the parameters that are going to come in liver function tests? I hope that clinical biochemistry comes in your final year paper. And they just asked, what are the different tests that you would do in a liver function test? And then they said, if urea level is going to be this much, does that person, what are the other things that you have to check in? The person was not able to answer. And they said that technical round, you have to really improve on your fundamental knowledge and get back to us. We will wait for you. So when you're reading papers, again, for scoring packs, for scoring marks, for scoring research, everything is there. But these are some of the areas that I personally feel that you should be not, if not strong, at least familiar enough. When they say that renal function tests, at least you should be say that, okay, I would be doing, my first test would be on a urea creatinine straight away. A liver function test, we would be doing on a SGOT, SGPT. A lipid function test, we would be going on a bilirubin, and then cholesterol, total cholesterol, HDLs, LDLs, HDL meaning good cholesterol, like that. Analytical chemistry, when they talk on, so centrifugation, chromatography, what is the principle behind centrifugation? What is the principle behind electrophoresis? What is the spec? All automated machines, it's all reading is given just like that, but you would be having some mechanism principle. The principle is nothing more than your spectroscopy. So what is a nanometer? What is the basic principle behind a creatinine? And then molecular biology and biotechnology, wherein we talk of DNA extractions, how extraction is going to be done, what is a DNA, what is a RNA, what do you mean by a cDNA, like that. So these are some of the few questions that I put on across in the technical round. And I feel that when you are getting into the lab diagnostics, you should be aware of the basic things that are happening in a clinical, subclinical biochemistry, at least, if not the analytical and the remaining area. So how to develop the skill set? This is where I said, how to develop your skill set during your college time. Use your vacation with a plan. Choose your training with a purpose. Not just because everybody is doing everything, you also don't go for that. You feel what is your strengths. You choose a plan for that and build it for your career development. Whatever you do, okay? Again, I would like just to get back to how ma'am was telling about me. She said that I'm more interested in doing a lot of online courses. That is true, ma'am, basically, because that helped me somewhere in my present situation, my present position, where I was able to talk with a lot of senior clinicians and senior officials, because they picked me out. They said that you have done this. We saw that you have done something on public health. You have done something with respect to research statistics. So we would like to get in touch with you. So whatever plan that you choose, whatever internship that you choose, whatever course that you choose, it should help your career directly. Don't choose anything which is not going to benefit you just for the sake of some X, Y, Z, or doing it or suggesting it. Doesn't mean that it is going to fit you. Each and every individual, you are going to have your own strengths. So understand your strengths, know your strengths, choose your training with a purpose. Not everybody can become a molecular biologist. So what? You get yourself expert in chromatography. If you're not able to do chromatography, get into spectrophotometry. Like that, you just get into that. Get the recommendation letters from your department, approach good labs beside which is near your college or near your locality, talk to them. And maybe this is one place where I would request all the colleges and the departments to really take an initiative, wherein if you have somebody who's got a good place, a good laboratory where they can allow your students to go and observe, have them as an internship, please encourage the students to go and do it. That will really help them a bigger length, a bigger distance when they are going for their career. And get a proper certificate and recommendation letter. There is nothing more eligible or more deserving than this. You can't just keep telling everybody that I have done this, I have done that, I have done this. This is what that will speak in your resume. We can see here as a student, if you go for an internship, If you go for a small course, and then if you go for a 1 month lab visit, you will have a certificate, and that is not going to be available with a lot of other students who are just not done anything. So when both of you are going for an interview, you would be preferred more than the person who doesn't have this. So this is what I mean by skill set. You plan for a good internship course. It need not be a lab course. It can be even an online course. If you have the benefit of going to a laboratory or hospital, go do it. That will definitely help you out in your long run, and get a recommendation letter from them. And also when you're leaving from your college, get a recommendation, get a certificate from the department stating that you have been doing like this. Get a letter of recommendation, a letter of appreciation, whatever it is. All those things will matter, because you don't know what your company is going to expect. And the last one, again, establish a good alumni platform and make use of it in the right way. It is not just to call them for parties, and not just to call them, say them hi, bye, it's been a long time since we met. If that person is going to be in a good organization, make use of them. Ask them whether we can come for a training, or ask them to intimate whether if there is an opening to our department, so that the department can select the right deserving person and they can be sent. So colleges will play a huge role in creating the alumni and alumni platforms, not for just get-togethers and yearly ones, but if we establish a good alumni platform, that will help a lot of the forthcoming generation of students. We might be there, we might just pass out, we might move on, but that platform will definitely help lots and lots of students. So this is again an initiative that has to be taken by the students after due permission from the department as well as the college, and make use of it in the right way. And this is again, all we keep telling you again and again, we keep saying that communication skills, it is more important. Surprisingly, when you see in pharma industries, right, you find that people would be excellent in their work. But when they come to present, when they come to write a proposal, when they come to write a project, when they come for a discussion, we will find that there are only very few who will be very dominant in their communication skills. Again, that lets you down by a considerable percentage. Imagine you have got a very good product that nobody else has right now found out. You are coming in and you are going to present it before a big gallery. 1,000 people are sitting there. If you are not having a good communication skill to keep that 1,000 people focused, listen to what you say, then you will become a big failure, because they will say that we are not able to understand what you are telling. So communication skill becomes a very important attribute. You might ask me, sir, I just do my work, I just switch on the instrument, I do it. What is there for me to learn? What is there for me to write? Why should I have a communication skill? But you should be able to talk about it. Somebody is going to ask you, someone, either of your friends or your juniors might ask you what exactly you're doing. If you're not able to tell them, you are misguiding them, you are letting them down. So then that way also it is very important. So it will learn to write procedures in your own words. We have records. I think faculty members will know that we pass on records for years. That is only to ensure that you're not making mistakes. But you should understand. You should understand what is the protocol. You just write the protocol, look into the protocol, frame your own sentences, write it. It will be very useful when you move high up on the ladder. Why I'm saying this is, initially you will be just doing what others are telling you. But at one point of time in your career, you will be there where you have to tell people what they are supposed to do. And if you are not able to communicate, how will they listen? How will they know what exactly you want? So communication becomes very important for you. And it will also help to establish a very cordial atmosphere. You talk to people, you keep the place quite peaceful and happy. People will be ready to help you if you're in a problem. You help people and get back help from them. That is very important. For that, communication skills are going to be very, very, very important. And these are some of the online courses that we talk on. There are so many government bodies like NPTEL, SWAYAM. I think people would have got familiar to it. Ministry of Human Resource Development, the Government of India have launched a lot of courses, online courses. You can enroll to them on free. If you want a certificate, you have to pay an examination fee. It varies from 12 weeks to 16 weeks to 4-week courses. There are so many courses. So for example, these are some of the courses that I myself have personally done in NPTEL. You can see here, biostatistics, health research fundamentals, cell culture technologies. And there are other private sectors also. They do a lot of online courses like a professional diploma in clinical research, advances in scientific writing. So these are some of the things that you can enroll on. Keep browsing, keep looking, be curious to know what is happening, enroll yourself. If you are able to pay, get your take the exam, get it done, it will definitely, see, again, these things will add to your CV. So you can now feel not just an MSc or a PhD or a BSc. You will have online courses, you will have internships, you will have training inside a hospital, you will have worked experience inside a lab, and then you approach a bigger organization. Definitely they will pick you out for the first round, and from there it is your expertise, your competence to move on further. This is for, again, this is a slide which my intuition made me put this, but I didn't know after looking at the participants. And in the beginning of the talk itself, I told you that the diagnostic field, you don't confine to genders. I have given you examples of 10 proud women of our country. So you can look into their designations here. Right now, we might think those people are big, but you should understand that these people exactly started their career where you are right now. In their own fields, in their own ways, they would have started their career exactly the position where you are right now at this point of time. Maybe it is exactly around 5:45 in the evening. You can see here, you know all these names, Biocons, Apollo, and all these things. But these people are inspiration for all of you. More than if this lecture is going to help you or not, if this talk is going to help you or not, I would be more happy if you're going to see these people and get your inspiration and seeing like, yes, we can also do it. The only thing that we have to do is get ourselves equipped, be smart, be proactive, be aware of our opportunities. Surely you will also become, if not like this, you will become more proud. You will make us more proud than this. And a few lines about my company, having told all these things. This is my lab. This is my company, Primegen Healthcare Laboratories Private Limited. We can see here we are a specialists. We are not a general lab. We do routine testing, but our specialization is going to be more on prenatal genetics, molecular biology, cytogenetics. We have got all these things in our lab. So we are more of a specialized lab, a specialized private lab. And we have been doing this till last month. And the COVID made us go in for an enable accreditation. And we said that we felt that we have to do for the society. And we got ourselves approved by ICMR, the Indian Council of Medical Research Lab, for COVID testing. And we are now doing COVID testing. And I have squeezed my 1 hour out from COVID, this testing time. And I'm so happy to talk. So we've come here. And COVID testing in a molecular biology lab, just to show a small, infuse a small light into it, how that COVID testing happens in our lab. We get the sample, a throat swab, and we do a extraction of RNA. And that RNA is going to be converted to a cDNA, and we put it in we put it with along with the other PCR mixtures, and we do a RT-PCR, real-time PCR. And the fluorescence will be picked up, and we find out a threshold value, the wave, the curve at which this is going to be picked up. And from there, we start detecting, we start interpreting results of saying it as positives and negatives and like that. That is a pretty interesting one, which we had started rightly right now. And it is going for 4 hours, 6 hours, 100 samples like that is going on. So this is my final concluding statement for all of you. Life always presents problems only when it knows that you have a solution. The solution you will find only when you're going to be hardworking and a smart worker. Automatically it will be there. It is there somewhere near you only. The only thing is you have to put the right hard work and you have to be very smart. So this is the carry home message that I have to say with respect to this 45-minute session that we have had. There are umpty opportunities that are present. But the only thing is you should not say that this is not for me. You should not say that this is too high for me to reach. You put in your hard work, you put in the right places at the right time, and you get yourself. The most important thing, be smart, be aware of what the situation is, be proactive, and definitely you all will be having a very, very, very shining and a prospective career. You can always reach me in this mail ID or this phone number for any further clarifications. And one more thing that we need to share on is our company, once this pandemic situation eases off and once we have the sort of transportation and make feel free for the students, we are going to launch in with a lot of internship and training programs in our lab itself with respect to general diagnostics or molecular biology as well as specialty diagnostics also. So we will get in touch with you through the corresponding authorities, and we would let you know also. In the meanwhile, if you want, you can always contact me in this particular mail ID, balavaskar.r@primegen.in or balavaskar@gmail.com with this mail ID. Okay, so that's it for the session, and I'm ready to take the questions.
Sir, there's a question from Palanisamy.
Yes, ma'am.
What about the prospects of zoologist in pharmaceutical companies?
What about the prospect of?
Zoologist in pharmaceutical companies.
You would be getting in pharma. It's again, it's a very expanding field. Zoologists, maybe once you get into a lab, there you would be definitely taken only as a technical technologist person, wherein your inputs would be coming in with respect to drug dosage, manufacturing, and all those places. Again, your role would be coming into play. That's why I said at the beginning itself, don't just narrow down your options. If you improve your skill set, if you have right skills along with this also, you would be definitely considered. See, all biotechnology or molecular biology or all the fields that have come up in the last 20 years, 30 years. So it is a ever developing field. So zoologist, pharma company, I will not say that there is no relationship at all. You might be involved somewhere in testing, definitely. Any other questions?
One more question, sir, from Varsha. What are the technical skills apart from the academic or needed in a resume?
Your technical skills, basically, the most important thing is you are having your own lab. In your syllabus, you have your lab sessions, correct? You need to understand the principle of all those things. And as I suggested, spectrophotometry, chromatography plays a huge role. I know people who have just mastered in GCMS and HPTLC who are earning much more, who are just having a very great careers. So technical skills, meaning you need to get into labs, get into handling. The real-time experience is what is more important for you. So basically, you have to approach through your organization, through your college, through your department. Only then those people also will be able to understand your interest in that. So you just go tell them, they might not be able to really say that, okay, they'll tell ability later. But if you go with the letter from your college or your department, then they will definitely help you out. Your technical skills will definitely be with respect to instrumentation. That is what I would suggest as technical skills. Next question, please.
A question from Logambal regarding COVID test.
Okay.
Regarding COVID test, after the virus moves to lungs, will the result be positive for swab test?
Right now, it should. That depends, madam. We cannot shortly say that real time. We need to do the test, and only then we would be able to really give a very confirmative statement on that.
Another question from Santosh, sir. Can you suggest the opportunities for biotechnology graduates?
Biotechnology graduates, that is there are plenty. All you have to do is you have to be good in your practicals. Your molecular biology is going to hold key. As usual, biochemistry will be definitely there. But if you're going to just focus yourself, your career only on being a biotechnologist, then I would suggest that you should be very strong in your molecular biology practical skills, extractions of DNA, running gel electrophoresis, and reading through your PCRs, handling different specialized PCRs like that. So that is where you have to then go in for a specialized laboratory, wherein you would be more absorbed as a molecular biologist. And right now, one other thing that we need to mention is we are all so focused on this COVID that we are talking only on COVID, but there are a lot of other infectious diseases and which could be done by a biotechnology, which could be done on a PCR. For example, we do it on HCV, we do for tuberculosis. TB IGRA testing is there. We do for tuberculosis also with respect to as a molecular biologist. So the opportunities are plenty. But if I say that you need to be having a competence, you should be on molecular biology to be more precise. Next question. Is there any other question?
Yes, sir. What role does biochemistry playing in the battle against COVID-19?
What role does biochemistry play in the
Battle against COVID-19.
See, you cannot actually uh, classify it as an individual subject based role as a biochemist or as a molecular biologist, as a microbiologist, how you can classify it. See, when you're going to say as a biochemist, you would be handling sample, how to handle a sample, right? This is not a joke. Handling a sample with respect to COVID-19, you could have seen it all in your media and all those things, the PP that has been worn, and how difficult it is for all these professionals to handle, to do the sampling and all those things. Those things that are being, you will understand. It is more of, understanding of like saying like, okay, as a biochemist, I know that I cannot just like that to do this, because if it gets infected, it will affect me. We are just not talking only on a COVID detected, not detected. If it is going to affect me, if I'm going to talk of a comorbid situation, see, for example, a person comes for COVID testing, they also come with other tests also, LFT, RFT, CBC, they do it. And we find that if the person is going to have a positive, you will find, are there any changes in the biochemical testing also? As biochemist will come into that role there. They can play a role there. They can interpret the values. They can see actually ICMR is now suggesting that we have to go on a lot of antigen antibody studies, check out what is the relationship between the cases and the IgM level or the IgG level, or right now they are expanding it into even get into CRPs, LFTs, see what are the other biochemical parameters that are changing with respect to detected positive cases. Are we finding any changes? So that is going to help in their treatment. So that is where biochemists will come into, since the question is going to be more oriented on what a biochemist is going to do. I feel that more than the testing, after the testing, if the person is going to be detected as a positive, then you have to get in with their other parameters. Are there any changes? Are they comorbid? If a diabetic person is being tested with the COVID positive, then what happens to their sugar levels? What happens to their lipid levels? Is there any impact on that? So now see, this COVID studies are growing. They are continuously being developed. There are so many updates. There are so many studies that are being, and that is why I told you, for the next 2 decades, life science graduates, you're going to you're going to just have so much of work to do, because every now and then they're going to say, see, now they'll say that, okay, we want to understand what is COVID-19. Molecular biologists came into role. Then they said we didn't have time to really find out, so we just went on for bioinformatics. We said docking studies will understand how the phytochemicals or the drugs are going to interact, and then they went on to have the animal studies. Vaccine development is going on one side. Now they said that during COVID, then during this virus, there is going to be the antibodies are developing after particular day. We want to know what is the rate. Get in with IgG, get in with IgM, go in for ELISA. And now they are telling that we want to know what effect is there with biochemical parameters. So now all the labs, they will start thinking on, okay, when a person is coming in, the clinicians will say COVID plus LFT plus RFT or whatever it is. We will see how it is changing. So we are all giving inputs. So we are contributing to this field. We are contributing to this particular human race at this particular generation of time, where our inputs are going to be very important, and they are going to give help in inferring in one thing or the other, in some way or the other, definitely. So yes, biochemists do have a bigger role to play.
And a last question, sir.
Uh-huh.
What is the reason why COVID test results vary from same person from place to place? That is from lab to lab.
That may be because of the testing, the type of testing that is going to happen, because there are going to be, to my knowledge, there are some 100 approved kits by ICMR which could be used for testing. The result will be the same. You will not have one case, 1% for 1% detected in one lab and not detected in another lab. It will never happen like that. But the kits that is being used, right, that might vary, but the final result will not vary.
Thank you, sir. Thank you for your more informative session, sir.
Okay, thank
May
you
I
very much,
invite
thank you.
Dr. Meenakshi ma'am to propose vote of thanks?
Yes, ma'am. Am I audible?
Yes, ma'am.
Respected speakers of the day, our most valued participants, and the organizers of the team. It's my privilege to propose the vote of thanks in this workshop. I, on behalf of the Department of Biochemistry, Bharati Women's College, extend my heartfelt thanks to the speakers of today's session, Dr. P.A. Abhinan and Mr. R. Balabhaskar, for accepting our invitation amidst their busy schedule during this pandemic situation and sharing with us their findings and opinions for the benefit of the student fraternity. As already mentioned, Dr. Abhinand has taken you to the world of bioinformatics in a precise manner and has clarified all your doubts. Thank you, sir. And the various avenues available in the healthcare industries have been unfurled by Balabhaskar in a simple and firm manner so as to reach each and every individual participant. Now, it's up to the graduates to identify their field of interest and upgrade themselves for their employment or to become an entrepreneur to reach their goal to greater heights. And with the permission of the speaker, Mr. Balabhaskar, I'm proud and happy to share that he has been student of Dr. Fatima in his undergraduate, student of myself, Mr. Dr. Meenakshi, in his PG level, and he has completed his research under the guidance of Dr. Vijayalakshmi and is about to get his degree. And the confidence in life is essential to achieve our goals. And our speaker, Mr. Balabhaskar, is a living example where he has leaped from the academics to the industry to serve the nation. Thank you, sir, for your wonderful lecture where you have unfurled the avenues available for the students to go on further. And I will be failing in my duty if I don't thank our research scholars, Shireen, Divya Bharati, and Samina, who have helped us throughout the programming of this workshop and made it a grand success. Thank you, speakers, the participants, for attending this workshop. Thank you, one and all. Dear
Thank you, ma'am.
participants, dear participants, the feedback link is given in the chat box. Please make a note of it. As we can allow only 100 entries per day, you need to again use the same link tomorrow evening to get your certificates. Me.
End meeting, end meeting, end meeting. Where is it?